Let's start with talking about nucleotide structure. So they're the monomers that make up nucleic acids. So they're the smallest subunits of things like DNA and RNA. So they're all comprised of a pentosugar, which is 5 carbons. And the way to tell apart ribose and deoxyribose is to look at carbon 2. If it's ribose, it's got an OH on carbon 2. And if it's deoxyribose, then it's only got a hydrogen, so it's missing the oxygen, which is where the name comes from. If you think about the way that they bond, which we'll talk about in a bit, using carbons 5 and 3 is 2 is really the only one that's kind of available to have anything different about it and still be able to bond the same. We also need to know about the different types of nucleotides. So they're separated into purines and pyrimidines. So the purines are adenine, where we'll use capital A, guanine, where we'll use capital G. They both have a double ring structure. You can see some pictures off to the far right on the page, but it's not visible on the screen right now. Pyrimidines, on the other hand, is thymine, which we use a capital T, cytosine, where we use a capital C, and uracil, where we'll use a capital U. Uracil is particularly of note because it's only found in RNA, so it's not found in DNA. All of them have a single ring structure, and they can be identified by looking for the Y in their name. So thymine and cytosine have a Y in their name. Comparing DNA and RNA, so this is just thinking about their nucleotides. So DNA contains deoxyribose, whereas RNA contains ribose, and DNA will contain the nitrogen-containing bases A, C, G, and T, so adenine, cytosine, guanine, thymine, whereas for RNA it will be A, C, G, and U. Looking next at the way that they start to bond together. So a really important term to be using here is phosphodiester bonds. So you can see it written there on the left-hand side. So condensation reactions form this bond, and hydrolysis reactions break this bond. You can see it indicates here in the picture what is included as part of the phosphodiester bond. So it's the carbon of that top nucleotide and the carbon of the bottom one and the phosphate and the bonds that link them together. So it's a kind of carbon, oxygen, phosphate, oxygen, carbon. Looking at phosphorylated nucleotides, so ADP and ATP, so they are ribose, which means that they will have an OH on carbon 2. It's always adenine that is there, and that's going to then be joined to a ribose. That's the adenosine. With ATP, it's then 3 phosphates, and with ADP, it's just 2 phosphates. So it's adenosine diphosphate is 2, and adenosine triphosphate is 3. We will be making a start with the topic of respiration and also photosynthesis in upper 6th, so we'll come across ATP synthase there. It's also good to have an idea of some of the different roles and uses of ATP, so active transport is always a good one, as is muscle contraction. Muscle contraction, that gets covered in more detail in upper 6th. So anabolic reactions as well. You might not be as familiar with the term anabolic reactions, but you'll know lots of examples of them. So it's any time where big molecules are being built up from their components. So for example, proteins being built from amino acids. Another example that works as well is cell division. If we look now at the structure of a nucleic acid, so we've got DNA, and it's important for us to know how they pair together. So thymine and adenine are always paired together with 2 hydrogen bonds. Cytosine, guanine always paired together with 3 hydrogen bonds. So A and T is 2, C and G is 3. A good keyword to be using here is complementary base pairs. So A and T and C and G are complementary base pairs. The next thing we can see here is to do with it being anti-parallel. So if you see the 3 here, so that indicates it being carbon 3 there at the bottom. And then it would be 4 and then 5 joining to the phosphate, and it would carry on up the chain, 3, 4, 5 phosphate, 3, 4, 5 phosphate. And so that top end ends with a carbon 5. So that's why it's got a 5 with a little symbol there. So that's 5 prime. And then on the other side, you can see that it starts with a 3 at the top. So it's 3, 4, 5 phosphate, 3, 4, 5 phosphate, 3, 4, 5 phosphates. You can see it's going in the other direction on the other side. So that's what it means for it to be anti-parallel. So they're not parallel going side by side. They are anti-parallel because one is the other way around from the other. Although this might seem like a slightly strange concept, it's actually something we'll all be really familiar with because it's exactly the same with roads. So different sides of the road being driven on, so we always drive on the left, but it's anti-parallel. Another key word for us then is the DNA double helix. So that's this three-dimensional shape where the 2 nucleotide strands, so polynucleotide, is then twisted as part of a DNA molecule. We also need to be familiar with practicals and how we can precipitate out DNA from a sample. So there's a few different steps, and generally the questions tend to focus on what the point is of some of the steps in the method. So the first step to this is about breaking open the cells, or cell lysis, so the cells being broken open, because you can't extract the DNA, can't precipitate or isolate the DNA unless you can actually break open the cell. So there'll usually be some kind of mashing, kind of using a pestle and mortar, something like that, or maybe blending it to break open the cell walls. Then you would use some kind of detergent, so washing up liquid maybe for that. And what that's going to do is it's going to disrupt the cell membranes. So that's going to disrupt the membranes that are between the outside of the cell and the DNA. So that's going to be releasing the DNA then into the solution. We also add salt as well because it helps with this process of getting the DNA out of the cell. The next steps that is worth us talking a bit about what the kind of function of it is. So filtering is important because filtering removes any of the kind of solid parts. For that, if you're using plant material, then there could be a lot of kind of larger solids that might need to be removed and filtered out. Really important, and one that gets asked about a lot, is adding protease enzymes. So we know proteases break down proteins, but we can be more specific than that when we're talking about this practical, because it's actually going to be breaking down the proteins that are associated with the DNA, or the proteins that are bound to the DNA. So that's the histone proteins in the cell. Okay, last steps then. So we make sure that we use ice cold ethanol, and you can see from the picture this technique of trying to kind of trickle it down the side of the test tube. So it's really important to pour the ice cold ethanol carefully and also slowly so that it forms a separate layer. So ethanol is less dense than the solution below it. So long as you are careful when you pour it down the sides, it will sit as a layer on top. The DNA is not soluble in the ethanol, but it will rise up into the ethanol above. And when it does that, it therefore precipitates. So it goes from being dissolved to moving into a solution where it's not soluble. So it becomes more of a solid. And so we would say that it precipitates as a kind of visible white kind of strands, kind of stringy-like structures. This idea of disrupting the cell membranes is really important. That comes up as well for the membranes topic, but it's not just the kind of cell surface membrane, it is also the nuclear membrane as well. But disrupting of the membrane is a really important term. Haven't seen a lot of questions where the function of the salt get asked about. But what it actually does is it is helping to kind of clump the DNA together. It's disrupting some of the charges that are present within the DNA, which causes it to kind of clump together differently. And just to emphasize how important it is that it is slow so that you do get that layer on top. Otherwise, you can be waiting ages for it to settle. Okay, moving on to DNA replication then. So 3 key enzymes here. So we've got DNA helicase as the first one. Now this picture's not perfect because it doesn't show exactly where DNA helicase would be acting, but it would be as part of this step here because the DNA helicase is going to be unwinding the DNA helix, and as part of that as well, it's going to be breaking hydrogen bonds between those complementary base pairs. Now, technically, there is another enzyme that is working alongside DNA helicase that's called DNA gyrase, but OCR don't ever really mention it, but you might see it sometimes if you are doing any further reading to do with DNA replication. So we give a little bit more credit to DNA helicase and we don't mention DNA gyrase as part of the course that we're doing. So our next enzyme then, DNA polymerase. Now this does something really specific. It is all about forming new phosphodiester bonds to form the new sugar phosphate backbone. So it's all about the formation of those really specific phosphodiester bonds. We will talk more in upper sixth about DNA replication, and particularly we talk more about mutations and the effect of mutations. But part of why actually mutations are quite rare is that each new DNA strand is created from an existing template strand. So that's the basis of semi-conservative replication. Now you can read about Michelson and Stahl's experiment of kind of supportive evidence for this theory and why we don't really discuss any of the other possible theories. But for this one, it's about using an existing template strand to make a new DNA strand that's then complementary to the existing templates. Now, mutations do happen. The spec specifically describes them as being random and spontaneous. There are a few different kinds of mutations, which, as I said, we talk a bit more about in upper sixth. So it could be that the bases go in the wrong way around, or they're inserted in the wrong order. It could be two are kind of the wrong way around. It could be that it's the wrong base that gets inserted. It could be that there's an extra one that gets added, or that there is one that's left out. Now, some of them are going to have a more significant effect than others. Think a little bit then about what the nature of the genetic code is. So the first one in here is about it being a triplet. So what that means, if we're talking about the idea of a triplet, is that three bases, so that's your A's, T, C's and G's, so three of those codes for one amino acid, so triplet because it's three. The next one on here is talking about it being non-overlapping, so relatively straightforward. The first three bases, then the next three, exactly the same as we read words, as we don't go backwards halfway through the previous word. Okay, this next term means something quite specific in the context of talking about the genetic code. So there are four different bases, A, T, C, and G, and a triplet contains three bases. That gives you 64 different combinations. You can work out the maths if you want, but it's 64 different combinations. There are not 64 different amino acids. There are, in fact, only 20 different amino acids. So there are only 20 amino acids and a stop codon. What this means is that there are some spares, but it wouldn't make sense for those triplets to code for nothing. So instead, there's a kind of doubling up. Now, the best analogy that I can think of for this is it's a little bit like synonyms with words, is that we can have multiple words that mean the same thing. If we use the example of valine, so the three letters that I've highlighted is the codon as it would be on the mRNA, but usually if you look it up, the codons that code for amino acids, it will instead list the three letters for the DNA. So with valine as the example, GTT, GTC, which is the one that is shown in this diagram, GTA and GTG all code for valine. So it actually doesn't matter what the third letter is, so long as the first two letters in the DNA sequence are GT. The universal nature of the genetic code, what that means is that the same gene will code for the same polypeptides. It will code for the same primary structure of the protein, the same sequence of amino acids, regardless of what species it is. The last part here then to do with transcription and translation. So I'm not going to go into loads of detail about transcription and translation. We're just going to focus on the enzymes and the structures that are mentioned at the top. So the first one being RNA polymerase, which is an enzyme, and it's RNA polymerase which synthesizes mRNA. Now that happens in the nucleus. Remember, the DNA cannot get out of the nucleus, but the ribosomes, which synthesize the polypeptide using that DNA strand, are in the cytoplasm. So the gene has to get out, and that's where the mRNA comes into it. So the mRNA is single-stranded. It is RNA, so it will be ribose. It will have uracil rather than thymine, and it is complementary to the coding strand or the template strand of the DNA. So it only needs to be a copy of one side of the strand because they are complementary. That's got all the information needed to code for the protein. And it's the mRNA that's going to be carrying the genetic code, the gene for whatever polypeptide is going to be synthesized from the nucleus out through a nuclear pore and into the cytoplasm, and it'll be taking it to a ribosome. Now, usually in these diagrams, they use free ribosomes rather than looking at the rough endoplasmic reticulum. The next one here, so tRNA, so these are the structures that are shown. That's a kind of simplified version of the structure as it is with a lot of these, but a tRNA will have an anticodon on one side that is complementary to an mRNA codon. So the three bases that make up the anticodon are kind of exaggerated. The entire tRNA is made of RNA, so there will be other bases there as well that kind of fold up to make the structure of the tRNA. On the other end of the tRNA, it will have a specific amino acid, and that will be the amino acid that is coded for by the codon that the anticodon is complementary to. So that makes sure that the tRNA brings in the correct amino acid to join to the growing polypeptide chain, so to form new peptide bonds and to form that growing polypeptide chain and to determine that primary structure of the protein. The last one then, so rRNA or ribosomal RNA. So this is what forms the structure of the ribosome. If you really think about it, is that you couldn't have the structure that synthesizes proteins made of protein. It has to be made of something that kind of comes before it. So ribosomes are made of rRNA or ribosomal RNA. The version of this page that you can see me writing in this video is available on OneNote, along with a related exam question from a recent paper for you to have a go at.